18 lecture on the regulation of gene expression. So we have two basics that we need to familiarize ourselves with, and of course there are going to be some subcategories within each of these, but there are two ways that we can control our expression. One is with physically controlling the gene expression itself, turning it on, turning it off. And the second is going to be regulating the protein activity that is the result of that DNA. So let me exit this, get it off the screen. So the Operon model, like it says on the screen, is always on the AP exam. You see the name of the scientists there. What's typically on the AP exam is not the nature of science, the scientists themselves, but typically you get questions about operons, and there are specifics that are often asked, specific operons that are tested over. Now notice that this is unique to prokaryotes. We'll spend some time on eukaryotes in a few minutes, but when we see an operon, they're going to be made of this operon area and a regulatory gene. So the operon, see, it's got some special pieces there. It's got a promoter, an operator, and then structural genes. So you'll see it's just kind of an on-off switch kind of thing for controlling expression. So here's an example. There's this protein that's a repressor that binds to the operator. So the operator is going to be where the switch is flipped, for lack of a better term, or along those lines. So when this repressor is bound, the RNA polymerase cannot proceed down the chain. So lactose is absent. So the repressor is active. The operon is turned off. So we don't want to always be breaking down lactose. And when I say we, I mean prokaryotes. Remember, this is specific to prokaryotes. So they don't want to waste a bunch of energy making the enzymes that are going to break down lactose if there's no lactose present. So that's where this repressor is going to bind to the operator and block the RNA polymerase from making that enzyme. So this repressor protein is the blocker. There's going to be the other portion upstream of that is going to be called the promoter, which is basically just an attachment sequence for the RNA polymerase to start transcription. And that might happen even if the operator is full. If there's a repressor present, attachment still might happen at the promoter. It's just going to be physically blocked from proceeding down the line. Like I said, the operator is going to be the switch. And again, if the operator is filled, then polymerase can't continue down the chain. We can't get our RNA strand created. The structural gene is the thing. So it's the enzyme that we're trying to make. If we're talking about lactose, for example, then the lactase is going to be the thing that we're looking for. That's going to be the structural gene. So I've talked about lac a few times. Lactose, we call this the lac operon. It's kind of our shorthand nomenclature for it. And that's going to, again, be to digest lactose. It's called an inducible operon. That's an important term. It's called an inducible operon because it only works when the substrate is present. So only if lactose is present will we be able to create the enzyme to break it down. So here's a picture. Again, you've seen this one. The operator is full. There's a repressor there. And so there's no lactose, so there's no breaking down. If lactose is present, then the repressor is going to bind to the lactose instead of the operator, which is going to turn on the operon and create the things to break down lactose. So here's sort of what this is looking, what this would look like. So allolactose is going to be a just slightly chemically modified version of lactose that's going to bind to that repressor and inactivate it. When that happens, now the RNA polymerase can proceed down and can create these enzymes that are going to break down lactose. Again, we only use it when we need it. So this is going to be, it's going to happen until there's enough lactose digested so that the repressor can become activated. If you try to picture what this would look like in reality, there are going to be several of these repressors floating around, and if there's an allolactose available, they're going to bond. They're going to chemically bind. When that concentration becomes low, the repressor is going to bind to the promoter just because it's not been inactivated. So again, the net result is the cell only makes the lactose when it needs to. The opposite and kind of the classical repressible ion is the Tryptophan. And so it's on unless it doesn't need to be. So the Lactophan is always off until we need it. The Tryptophan is on until we don't need it. That's sort of what I said there. And it's the same. The picture is not drastically different here. We've still got an operator. We've still got a repressor. But the repressor is inactive until we don't need it anymore. We activate the repressor and it turns off the Tryptophan. And so notice that this is not breaking down tryptophan. This is building tryptophan. So that's a common theme to repressible ions. They're going to be things that we're building. So we're all, and I keep saying we, and I apologize about that. When I say we, for this segment, I'm talking about prokaryotes. So they're always making tryptophan until they tell the cell not to. And they're never breaking down lactose, and they're never making those enzymes to break down lactose until they need to. So if it's repressible, it's probably going to be catabolic. If it's an inducible, it's probably going to be anabolic. So here you see the mechanism when tryptophan is present. When there's enough tryptophan around, it's going to activate the repressor, and we won't need more tryptophan to be made. So again, it's concentration dependent. These are examples of feedback inhibition. It's going to keep the substrate at a constant level. When the levels drop, the repressor won't be effective, won't be active. So that'll allow us to control the concentration. So all that was prokaryotic. Make sure you're comfortable with those operons, the structure of them, and then kind of the feedback inhibition aspects of the two different kinds. When we transition to eukaryotic gene regulation, of course, it's always more complicated. You need to be able to make sure you can talk about several of the mechanisms in some detail. You need to be able to do more than just identify if we're talking about verbs with this stuff. So we can have control through our chromatin structure. Histone modifications, DNA methylation, and epigenetic inheritance are all going to be things that are involved in the structure of our chromatin. Remember, when we talk about chromatin, we're talking about how the DNA is an interphase, which is most of the cell's life. So get the histones, think back. It's been a long time since we talked about this, but once upon a time, I demonstrated how chromatin gets wrapped into chromosomes, and I wrapped a rope around my leg, and I wrapped it up, and it came around my arm, and I tried to use that as a model for how chromosomes are wrapped. In that demonstration, my leg is a histone. Histones are the things that are going to wrap the chromatin and eventually stack up on top of each other and eventually form our chromosome structures. But those histones aren't absent in chromatin. They're still there. They're just the DNA is wrapped more loosely around them. So if we attach, or if these acetyl groups are attached to the histones, the DNA is going to loosen up in some spots. If it becomes more loose, it's easier for the polymerases and the different enzymes involved in transcription to access that DNA. If the DNA is held really tight to the nucleosomes and held really tight to those histones, then it's going to be physically blocked off. You just aren't going to be able to get to it to build the DNA off of it. And here's kind of a picture. You see unacetylated histones, we get bunching. when we acetylate them, they spread out. The acetyl groups repel one another. So things become more available for transcription. Methylation is really similar, same kind of process. We're going to add methyl groups to the DNA bases themselves, and that's going to shut down transcription long term there. You see Barr bodies, that's an example that we've talked about with respect to the X chromosome. We shut down one of those. Think about the calico cats discussion that we talked about. I think that was before winter break. So that's one way that we can control expression. We can just turn it off for good. Genomic imprinting, we talked a little bit about that, about how this methylation pattern resets every generation. So you get the male version, the female version of the methylation pattern. And then last but not least is epigenetics, which is a really exciting field of genetics that's really developing and has really developed over the last 10 years. So it's kind of some of this modern genetics, and we will learn more about that in the near future. But it's the idea that our genetics change over time. We inherit what we inherit, but if you look at identical twins, they're identical at birth. But over time, sort of the nurture version of their phenotypes, their methylation patterns are going to change based on their exposure. Maybe one twin is a smoker, one twin is not. Maybe one twin works in a place where there is more radiation, or maybe one twin moves to an equatorial environment or something along those lines. Their methylation patterns are going to change, and they'll become less alike with age. If you see older twins, older than elementary or high school or middle school, like senior citizen twins, you'll see that they look pretty different. They don't necessarily look the same, and that's because their methylation patterns have changed over time. So that's sort of the first round of eukaryotic gene regulation. The next step is to look at transcriptional control. Can we do what prokaryotes do with operons? And the answer is sort of. We just do it in a different way. Of course, we have a common ancestor, so we're going to do similar things. We're just going to do them in a slightly different way. So I'm going to leave off the first video here, and I'll pick up the next video on the next slide.